Can you hear me?
Yes, doctor.
Okay. Thank you. Thank you very much. So today's lecture is about tubular secretion. I will share my PowerPoint. Tubular secretion. Bismillahirrahmanirrahim. So as you know, the process of formation of urine it's having three processes. Process number one is filtration. We discussed in the lecture of glomerular filtration. Let me make it full screen. Okay, so we have got these are the objectives of today's lecture, that we will be discussing the definition of tubular secretion, then role of tubular secretion in maintaining the potassium concentration, which is very important for the membrane potential for the excitable tissues. That's why the concentration of potassium has to be regulated by the body in a very critical way. And we will see what are the mechanisms of tubular secretion of different material ions like potassium, hydrogen, or inorganic acids, or organic acids or organic bases. So as I told you that process of formation of urine requires three processes. One is filtration, the one which we started with our block, that is glomerular filtration. So here is the glomerulus and efferent and efferent arteriole. It is filtered here. This process is known as the first step, that is glomerular filtration. Then once it is passed through the tubule, renal tubule, it has got various parts. In this one is proximal convoluted tubule. So things which are required for the body is reabsorbed. They are reabsorbed and added to the blood. They are back added to the back into the blood. So this is called as reabsorption. But some materials, some chemicals which are not filtered, because it is filtration rate is about around proportion. The portion of the blood which is filtered is only 20%. 80% goes here only directly to the peritubular capillaries. So it is not freely filtered. Only 20% will come here, but 80% will have the same amount of the waste products. Suppose there are waste products. So how they can be removed from the body? That process is known as tubular secretion. So they will be added back into the filtrate. Here is the filtrate. Now, once filtration occurs, it will be filtrate here, and it will be reabsorb those essential things like glucose, water, sodium, calcium, phosphate. All these are which is required for the body are reabsorbed. But those things which are not required by the body, what is not required for the body? Excess quantity of hydrogen, or excess quantity of potassium, or excess quantity of urea or ammonia. They are not required by the body, but they are just only filtered, maybe very little amount, 20%, and 80% again coming back into the blood. So from the blood, they has to be eliminated and added into the filtrate by the process known as secretion, tubular secretion. So in today's lecture, we will be talking about the process called as tubular secretion. What is the definition? It is written here that transfer of materials from blood into the tubular fluid. As I told you, this is a peritubular capillary, and here is the renal tubule. Okay, so things are coming from blood into the interstitium, then from the interstitium into the filtrate or the tubular fluid. This is called as This is called as tubular secretion. Why it is required? It requires mainly to control or regulate the pH of the blood. pH means hydrogen ion concentration in the blood. What is the normal pH of the body? The blood? What is normal pH of the blood or body?
7.4.
Are you listening to me? Are you with me?
7.4.
Yes, doctor.
7.35 to 7.45.
pH of the blood is 7.4. So it has to be maintained at 7.4. Also, to eliminate those substances which are not required by the body, we have to do, or the kidney has to secrete them. So that's why secretion is important. So what are the steps? The first step in the secretion is a simple diffusion from peritubular capillaries into the interstitial fluid. As I shown you in the previous picture, this is a peritubular capillary, and here is the, let me change the color of the pen, that's why, so that you will understand which is peritubular capillary and which is the tubulary. No, I will make it black so that it will be easily visible. So Bowman's capsule, proximal convoluted tubule, loop of Henle, descending and ascending limb. So first step is from peritubular capillary into the interstitium. This is simple diffusion. From there, entry into the tubular epithelium, which can be an active or a passive process. Here is the tubular epithelial cells. So it will enter into the tubular epithelium, and from the tubular epithelium into the lumen, This is the third step, second step, and first step. So three steps are there. First, they have to leave the blood vessel, that is peritubular capillary, come to the interstitium, then from the interstitium into the epithelial cells, and from the epithelial cells, they are released or secreted into the tubular lumen or the filtrate. Examples of things which are secreted are potassium, hydrogen ion, organic acids and organic bases, and ammonia. These are the main examples of secretion. So in general, we can say secretion is equal to this, the one which is coming here. Secretion is equal to total, that is excreted amount minus the filtration amount. So it will be here. So secretion is equal to excretion minus filtration. What are the common things which are secreted? These are hydrogen ions, potassium, ammonia, organic acids, and organic bases. They are organic acids and organic bases. They are formed in the body, or maybe due to some metabolism, or maybe due to food additives. They are coming into our body, and it has to be eliminated. So what is the importance of tubular secretion? Why it is important to add the things into the tubule even though after it is not filtered? It is important for disposing of the the things which are not required by the body, not already filtered. Something are filtered, can be easily going into the circulation, but some materials, they are not filtered, but still we don't want it. So it has to be added to the filtrate by a process called as tubular secretion. Then eliminating the undesirable substances such as urea, uric acid, we don't require, but still they are circulating in the blood. So we have to remove it by the process called as secretion. Then getting rid of the substances like excessive potassium ion. Potassium ion is very, very important element for what? For the membrane potential, for generation or maintaining the resting membrane potential. But if you remember, potassium is present in intracellular fluid, and it is also present in extracellular fluid. or extracellular fluid, or you can say plasma. But the quantity of potassium is maximum inside and minimum outside. And when we eat or drink some drinks, then it may change the extracellular fluid concentration, which is disturbing the resting membrane potential and also disturbing the excitability of the excitable tissues like nerve and the muscle. Muscle can be either cardiac or smooth or skeletal muscle. So that's why it will cause the problem in excitability of the muscle contraction or the nerve stimulation. One of the important thing of tubular secretion is it is required for maintaining the controlling the blood pH by causing secretion of hydrogen ions. Hydrogen ions are continuously produced in our body, and it has to be regulated with a very critical level. That's why regulation of pH is important. 7.4 is the normal. It may range between 7.35 to 7.45. The most important substance which are secreted by the renal tubule are hydrogen ion, potassium ion, and organic ions. Hydrogen ions are required for regulating the acid-base balance, and it is secreted in proximal convoluted tubule, distal convoluted tubule, and collecting tubule. All three places hydrogen is secreted. Whereas potassium, it is required for keeping the potassium concentration at an appropriate level. What is the normal potassium level in the plasma or blood? What is normal potassium level? Is it very high? How much is the normal potassium level? I told you in the previous lecture also.
3.5.
So potassium is secreted only in distal and collecting tubule, whereas other places it is reabsorbed. So DCT and CT, these are the two places where potassium is secreted. And again, it is not secreted alone. It is with the help of or with the action of enzyme called, hormone called as aldosterone. So aldosterone has a role in regulating the potassium ion concentration. Aldosterone. Organic ions, they are also secreted in the renal tubule. Their secretion is accomplished more efficient elimination of these foreign organic ions from the body, and they are secreted only in proximal tubule. Only in proximal convoluted tubule they are secreted. Here is the the importance of regulating the potassium or secretion of potassium by the kidney, because 98% of the potassium is placed in the intracellular fluid, whereas 2% only present in the extracellular fluid. So kidney is regulating this 2%. Kidney is regulating these 2% only. So regulation of extracellular fluid potassium concentration is one of the function of the kidney. In the ICF, it is about 150 milliequivalent per liter, and in the ECF, that is plasma, potassium concentration is about 4.5 milliequivalent. 4.5 milliequivalent per liter. When we say potassium balance, that means we have to balance the intake, how much we are taking, to the output, how much we are or how much the kidneys are giving out the potassium. This is important because we have excitable tissues like muscles and nerves. In the muscles, the cardiac muscles are very, very important. The cardiac muscles are very important because even increase or decrease in potassium will lead to arrhythmias. Will lead to arrhythmias or decrease excitability mainly. Okay. Again, this same slide, this slide is showing you what is the importance of regulating the potassium, because potassium is a very important ion for regulation of the excitability of the tissues. Both increase and decrease plasma can change the intracellular or extracellular potassium concentration gradient, and that's why it will change the resting membrane potential. So increase also and decrease in potassium level also is not good for the body or not good for the excitable tissues. That's why it has to be regulated in a normal levels. Most important one is the impact of potassium level is on the heart. It causes the decreased excitability of the cardiac muscle. Rise also and fall in potassium also both causes the decrease in cardiac excitability. Rise in potassium concentration decreases the excitability of the neurons also and the skeletal muscle also. So this will lead to weakness of the skeletal muscle. If the person is having excessive potassium ions, there will be muscle weakness is found in that person, and also maybe having a symptom of diarrhea and abdominal distension. So this is a diagram showing you the various places where potassium is reabsorbed and secreted. So as you know, the proximal convoluted tubule, mainly there is reabsorption. In PCT, almost 67% of the potassium is reabsorbed along with water and along with sodium also. In the thick ascending limb of loop of Henle, 20% of the potassium is reabsorbed. Again, it is reabsorbed here. So 67 plus 20, how much? 87. And in the last part, like the DCT and CT, there will be variable amount of potassium reabsorption or secretion. In the last part, that is DCT and CT, there will be a variable amount of sometimes reabsorption, sometimes secretion based on how much the person is eating, dietary potassium, how much you are eating. If you are eating more, there will be secretion. If you are eating less, there will be reabsorption. Also, it depends upon the level of aldosterone secretion. Aldosterone secretion has got an effect on potassium secretion. If aldosterone level is high, it will lead to secretion of potassium. Also, it depends upon acid-base balance, or you can say hydrogen ion concentration. Hydrogen ion concentration can also lead to change in the amount of potassium secreted by the collecting tubule or distal convoluted tubule. Also, it depends upon the flow rate, how much or what is the quantity of fluid or filtrate is flowing per minute. So flow rate will also decide whether the potassium is secreted or absorbed. And amount of luminal anions, negative ions, amount of luminal anions. All this will lead to regulation of the potassium in the late DCT and CT. Whereas excretion rate, based on these conditions, the excretion rate may vary from 1% of the total intake to 110% of the intake. All will be excreted plus added amount 110, 110. So it is a wide range. Sometimes it may be only a very little amount is excreted or given out in the urine. And in some cases, it may be all is given out plus secreted extra. Okay. And all it depends upon these factors. So you must know the factor. How much intake of potassium in your diet. Then what is the level of aldosterone? And it depends upon the sodium levels. What is the acid base status, whether it is acidosis or alkalosis in the body? What is the flow rate? How the urine flow rate is there? That means more formation and more flow rate of urine. And how many negative ions? The percentage of negative ions or presence of negative ion in the lumen. All this will decide whether it will eliminate 1% or 110%. Okay. For that, you have to remember two cells in the DCT and CT. Two different types of cells are there. One is known as special cells. These are called as alpha intercalated cells, and principal cells. These two cells are deciding whether the potassium is reabsorbed or secreted. So responsible for the distal tubule and collecting tubule are responsible for adjustment of the potassium excretion. How much should be excreted? It can be 1% or it can be 110%. What alpha intercalated cells will do? Alpha intercalated cells are responsible for absorption of potassium when potassium in the person the diet is low. When person is eating low potassium diet, then potassium will be absorbed by the alpha intercalated cells. Whereas there is another type of cell is present in the DCT and CT, which is known as principal cells. These principal cells, what they will do, if the person is on high potassium diet, then it will be excreted through principal cells. So alpha intercalated cells are responsible for absorption of potassium, and principal cells for secretion of potassium. The magnitude of potassium excretion is variable depending upon the diet. Also, it depends upon aldosterone level, acid-base status, and the urine flow rate. Here is the example of how the potassium is reabsorbed. So if it is reabsorbed, which cell it will be? It will be alpha intercalated cell. Alpha intercalated cells are responsible for reabsorption of potassium. And you will observe that this is the luminal side. This is lumen side. This is the interstitial side or basolateral side. You can say basolateral membrane. So on the luminal side, you will find there is a counter transport mechanism is happening where potassium is reabsorbed in exchange with hydrogen. So this is the function of alpha intercalated cell, absorption of potassium and secretion of hydrogen ion. So you may be thinking that it is mainly related to the acid-base status of the body. When the body is having or the urine is more acidic, then hydrogen ions has to be secreted. In replacement, potassium will be reabsorbed. Principal cells in the late DCT and CT, what they will do, they are under the influence of aldosterone. This will lead to more secretion of potassium ions. So potassium ions are secreted under the influence of aldosterone through principal cells. The level of or the amount of potassium secretion depends upon the size of electrochemical gradient. What is the meaning of electrochemical gradient? What is the difference between electrical charges in the lumen with respect to the inside of the cell, inside of the epithelial cells? So because there are positive and negative charges placed at both places, the difference between them is known as electrical gradient. Then the potassium here and sodium more here, or maybe different chemical ions, it is known as the difference is known as chemical gradient. Together with the charges plus the chemical nature of the particle will create a electrochemical gradient. Which will decide whether the person will secrete potassium ion or reabsorb potassium ion. So high potassium diet will cause concentration of these potassium inside the cells. Thus, the principal cell will increase the electrochemical gradient and inside the principal cells. This is all together in one area. High potassium diet will lead to concentration inside the principal cell, will cause increased electrochemical gradient, and this will lead to secretion of potassium ions. So you have to remember the role of aldosterone in regulating the potassium ion, role of aldosterone. Basically, we have learned that aldosterone is mainly responsible for reabsorption of sodium. But one of the factor, when let's say sodium concentration decreases, then aldosterone will be released, increase aldosterone. This is one method. Another method is if potassium is increased in the body, there will be increase in aldosterone level. So aldosterone will cause absorption of sodium by the principal cells. This is acting on the principal cells, which will increase the synthesis of more sodium channels at the basolateral membrane and sodium potassium channels at the basolateral membrane. It increases the number of sodium potassium channel, ATPase channel. So more sodium is pumped out of the cell, simultaneously more potassium is pumped into the cell, thus increasing the electrochemical gradient for potassium across the luminal membrane, which will lead to secretion of potassium ions. I will show it here. Okay, so two mechanisms are there for stimulation of aldosterone. Can be by potassium high level or could be because of low sodium levels. So either low sodium or low extracellular fluid volume or low arterial blood pressure lead to increased renin, lead to increased formation of angiotensin 1, which will be converted into angiotensin 2 in the lungs. And then it will come to the renal tubules to cause increased reabsorption of sodium, and urinary excretion of sodium will be decreased. Similarly, another way of stimulating the aldosterone secretion is when potassium concentration increases in the body or increase in the plasma. This will lead to increased aldosterone secretion, which will cause increased secretion of potassium and increases the urinary excretion of potassium. So as I told you, it may vary from 1 to 110%. That means there may be effect of aldosterone at this time to lead to increased release of or increased excretion of potassium. So two ways are there to stimulate the aldosterone secretion. One is by decreased sodium, or another way by increased potassium in the plasma. Same thing is shown here. This is about the principal cells. What is the function? Only thing is that you remember this is the opposite. Lumen is here. Lumen is here where the filtered potassium and sodium is going. And this is the basolateral size. This one is basolateral membrane. All other pictures we have opposite. Okay, so just be sure that when the aldosterone is released because of either decreased sodium or increased potassium in the body, this will, as a aldosterone, it is a lipid hormone. It will enter into the cell and it will cause more action over the nucleus or over the DNA, and this will lead to more formation of these channels across at the basolateral membrane, which will lead to more potassium coming in, and on the luminal side it will insert potassium channels, which will release the potassium into the lumen, and then potassium will be lost into the urine. Okay. So this is how, and simultaneously at this time it will be absorbing more and more sodium. So sodium reabsorption along with potassium excretion is the function of aldosterone on principal cell. So the end result is increased absorption of sodium by the principal cells and increased release of or secretion of potassium by the principal cells. Also, aldosterone has got effect on another type of cell, which is known as alpha intercalated cell. Alpha intercalated cells, which will lead to increased secretion of hydrogen ion. Increased secretion of hydrogen ion. So when the person is on high sodium diet, high sodium or high salt diet, so more sodium will be delivered to the principal cells. So more sodium will be available for sodium potassium ATPase pump. Then more potassium is pumped into the cell, which will increase the driving force for potassium secretion. Driving force for potassium secretion. That means when high sodium is there, high sodium will be absorbed, lead to secretion of potassium ion. Now comes the role of diuretics in regulation of the potassium. What are diuretics? Diuretics are drugs which will increase urine formation. How they form urine, excess urine? Because they may have excessive solute in the filtrate, and this will lead to the loss of water along with it. So there are different categories of diuretic. One is known as loop diuretic. What is mean by loop diuretics? Loop means loop of Henle. Loop means loop of Henle. So those diuretics which are acting on loop of Henle. In loop of Henle, there is descending limb, then there is ascending limb. In the ascending limb, there is a thin and thick segment, right? In the thick segment, you have got a channel or a protein which is known as sodium potassium 2 chloride channel. Sodium potassium 2 chloride channel. These loop diuretics will cause inhibition of this channel, sodium potassium 2 chloride channel, which is responsible for absorption of sodium, absorption of potassium, and absorption of chloride. If it is blocked here, this excessive sodium will go to the distal convoluted tubule and collecting duct. So excessive sodium, excessive potassium, and chloride. When more sodium is there, it will lead to principal cells will cause absorption of sodium, and excretion of potassium. So there will be loss of potassium there. There will be loss of potassium. Similarly, thiazide diuretics. These thiazide diuretics are acting in the distal tubule, distal convoluted tubule, which is blocking the sodium channels. They are blocking the sodium channels. And again, this will lead to increased sodium in the distal convoluted tubule, which will lead to absorption of more sodium and in replacement excessive secretion of potassium. So loss of potassium in the urine, more potassium in the urine. These are the functions of diuretics and how they are affecting the potassium concentration. So for that purpose, there are different specific diuretics are used to treat the congestive cardiac failure or blood pressure, or when you want to regulate the blood volume or the plasma volume, then you have to use some drugs which are not changing the potassium concentration, or potassium sparing diuretics can be used. Okay, the other function of secretion is to get rid of from the organic anions and cations. Organic anions and cations. Cations means positive ions, anions are negative ions. So these organic anions and cations, like examples are prostaglandin, epinephrine. After their action, because once they are released from their source, they will act on various cells. Once they perform their function, they may be moving in the blood and may cause increased action, or may cause the action to be for longer time. So for that purpose, they have to be removed from the kidney or from the body through the kidney. This is one of the way, that is secretion. So it will be secreted back into the renal tubule, and then it will be removed from the body. So like examples are like prostaglandin, epinephrine. After their action is done, they are removed from the body by secretion. Even some non-filterable organic ions are also removed by this process. Some of the food additives are also removed from the body by secretion, or some non-nutritive substance, and also the drugs. After the drugs, their actions is over, or some of the antibiotics, they will be removed from the renal tubule by the process known as secretion. One of the example of secretion is para-aminohippuric acid secretion. Para-aminohippuric acid is nothing but a organic acid used for measurement of, we have seen in the clearance chapter, renal plasma flow. Renal plasma or renal blood flow is measured by injecting a substance known as para-aminohippuric acid. So it is filtered as well as it is secreted. So that's why its concentration is more. We have given 600 is the clearance rate, whereas GFR is 125 only. So pH transporters located in the peritubular membrane of the proximal tubular cells. There are parallel secretory mechanism for secretion of the organic bases like quinine and morphine. So organic acids and organic bases both are removed from the body by the process of secretion. Another example of secretion is urea and uric acid. Urea, which is a end product or byproduct of metabolism of protein. It is freely filtered and 50% is reabsorbed in the proximal convoluted tubule, and then 50% is secreted, and then it will be removed from the body. So actually it is 50% is lost from the body. Urate are nothing but the salts of urea. Urate are nothing but the salt of urea. They are also freely filtered and they will be removed from the body by the process of secretion. PCT, there is reabsorption and secretion take place in PCT, that is proximal convoluted tubule. In the initial and mid part of the proximal convoluted tubule, reabsorption is more than the secretion. So in the earlier part, it is more reabsorption, and in the late part, it is more secretion. In the distal portion of the PCT, there will be secretion of urates, that is the salt of urea. Salts of urea are secreted. This is all about, this is not a big lecture, all about the process of secretion. Are you with me?
Doctor.
Yes, doctor.